All right, we'll go ahead. Is that started? Yes. We'll go ahead and start. I'm Paula Cushing. I am the advisor for the speaker today. Goran Shikak joined my arachnology lab in 2021 as a master's student at University of Colorado, Denver, where he's going to receive his degree, where he will receive his degree this Friday. That's when he's defending. And I'm pleased to say that Goran was also awarded the College of Liberal Arts and Sciences Outstanding Graduate Student Award at the University of Colorado, Denver, where he's receiving his degree. And I also serve as adjunct professor over there. So today, Goran will be giving you an overview of half of his thesis project. His thesis included two major projects: an evaluation of rehydration techniques to reconstitute alcohol-preserved specimens for which the alcohol had evaporated, and that work has resulted in a manuscript that is currently in review with a scientific journal. His co-author on that project is our own Jenna Anderegg, who is somewhere in the audience. So Goran and Jenna are co-authors on that chapter. His other chapter, which he's presenting today, involved using various analyses to revise a taxonomy of a group of camel spiders that are found in the Great Plains and the Chihuahuan Desert. And it's that work that Goran is going to present to you. Because he's defending on Friday, I would ask the audience, if you have some feedback, if something's not clear, please feel free to give him some suggestions at the end, ask some good questions, because the committee is going to be asking him a lot of questions, so he needs practice with that. So without further ado, welcome, Goran.
All right, let me see. Can everyone hear me? Yeah. Awesome. So before I get into the details of my research for this chapter, I would really first like to give special thanks to people who are very formulative on this project. Those who mentored me, like Dr. Paula Cushing, as well as Jack Brookhart, who I hope will be there at my defense on Friday, but also current and past lab members like Ryan Jones and Dr. Erica Garcia. They have been monumental in pushing me to be a future scientist, helping me devise my techniques and think through this project. Without them, I don't think I could overcome this insurmountable task. Beyond that, I'd also love to thank my committee, who I will get to see and be grilled questions on Friday, because they have been also very foundational for helping me develop my writing and my science. With that said, when I joined the lab, I got the opportunity to finally work on what I've always wanted to, something that is understudied and unknown as an animal taxa. And I got this chance to work on quite an enigmatic and unknown arachnid called a solifuge. Solifuge, colloquially known as camel spiders, are enigmatic nocturnal arachnids distributed across most arid and semi-arid habitats. And only recently have we uncovered with molecular work that there are quite some issues with their taxonomy and their evolutionary history, their relationships. In our lab, we focus our work within one North American family called Eremobatidae. And much of the work ongoing right now between myself and Ryan Jones is in the genus Eremobates. Recently, now that we know that there's an unresolved taxonomic issue, we're trying to focus our work to revise taxonomy of groups piece by piece, but there is still an issue of having diagnostic characters. Within Eremobatidae and the different groups therein, there are very few diagnostic characters, and so this work is trying to establish new characters that we can use to revise their taxonomy. Solifuges primarily rely on the chelicera for mating, and that's true with males, and so we see quite a unique array of features and shapes there. In 1951, the late Dr. Martin Muma used traits associated there and the patterns to revise the taxonomy of Eremobates, and in that, he helped to devise different species groupings. This is a large specios group with nearly 90 species. And so he looked at the morphological similarity and the biogeographic pattern to devise these groups. Unfortunately, for a lot of these groups, they had very few characters to help support them. In the case of the palpisetulosus group, one of the largest ones, it is actually a single character that was used to try to lump these taxa together. That character is called a retrodorsal process. So on these male chelicera, their jaw-like mouth parts, there's a small spur that all males share on that top finger. And so that character he used to lump together those 42 species into that group. Along with that, he looked at females that he collected with these male individuals and found that typically they had these triangulate-shaped genital plates depicted over here. So there were very limited characters that was used to lump together all of these taxa, but he worked hard to try to recognize what morphological patterns he could best use to sort this group. In addition to devising the group, he later created series owing to the large collection of species here. However, as we now know with recent work, particularly by Dr. Paula Cushing, there's not a stable diagnostic character for this group because it's not monophyletic anymore. In 1988, the late Dr. Muma and Jack Brookhart had worked to try to use morphometrics, measurements and ratios of the body, for the group to try to resolve the taxonomy, as they recognized that this was largely becoming a problematic group. They moved away from traditional leg and propeltidium, the head segment characters, to try to delimit species, as they found that it pretty much was mostly identical for all these different taxa. Instead, they focused their ratios efforts on the chelicera, those jaw-like mouth parts. They evaluated different components to try to test what was a species in their manuscript. And they found very few traits that they could reasonably use. However, there were some issues within that time. They feel the diversity that they put out may be overestimated because at that moment, they had very few samples to work with. They were limited to a handful in their best cases. In the worst cases, they had only the type specimen for which to evaluate. Those issues really came to light in the taxonomy in 2015 when Dr. Paula Cushing et al. Put out the first exemplar family phylogeny. And what this found for this species group in particular was that multiple clades were recovered across the phylogeny. It's not monophyletic, it's not coming out together. And so that uniting character used by Muma, this retrodorsal process, is not a reliable synapomorphy in itself. Instead, we had several clades recovered with biogeographic signature, highlighted over here with these markings. Interestingly enough, these biogeographic clades did actually somewhat fall in line with Muma's series designations that he put out with Jack Brookhart in 1988. Those are the nodularis clade for nodularis species, the Ahuanis clade, the palpisetulosus clade, and a mixture of two series in one monophyletic California clade. The first one we're going to focus on in this research is what we call the nodularis species. He created this series for 3 species, potentially 4, that are distributed across the Chihuahuan desert, and looked at how morphologically similar they were to separate this group out. Next is the palpisetulosus clade, and this is much broadly and wider distributed than the nodularis group. This is spread across the Chihuahuan Desert, the Great Plains, but also Colorado Plateau and Arizona Plateau habitats. And this was constructed originally for 8 species that were morphologically similar. The last, and also morphologically similar to that palpisetulosus group, is the Ahuanis clade, or the Ahuanis series. And that was constructed by Muma and Brookhart for 3 species they identified in 1988, but they only found these species at the time in the Sonoran Desert and Madreine Archipelago habitats of Arizona. While there is also California taxa involved within the palpisetulosus group, that is over 20 species that will need to be resolved in the future. This work is particularly focused on those biogeographic clades in the Chihuahuan and Great Plains. Following Dr. Paula Cushing's work in 2015, what we found is that the Eremobates palpisetulosus group has lost support, but we have a foundation to work forward to try to revise the taxonomy. We don't have characters currently that we can use to reasonably diagnose species, as well as differentiate these different clades. And so to resolve this, my approach was to use integrative taxonomy, look at not just the genetics, but combine that and leverage it with morphology as well. For the genetics, I'm particularly interested in what are the systematics for the focal taxa for these Great Plains and Chihuahuan species. To do that, we can use the UCE, ultra-conserved elements of DNA, to try to resolve evolutionary relationships particularly well for museum and degraded samples. And then to test species hypothesis, we can use the CO1 barcode gene, which in many papers, particularly for arachnids, has been found reasonably to work well in species delimitation efforts. But beyond the genetics, establishing genera and testing species, we also wanted to see if we could find synapomorphies for the proposed genre, these distinct clades. And so we focused on male and female genitalia characters and used morphometrics to generate ratios to see if we could reasonably distinguish them. And then further than that, could we find stable diagnostic characters for species? So to address the taxonomy and try to resolve this group, our goal was to sample as many of the species, which it's 15 among these clades, that we could. And that has involved working here at the Denver Museum of Nature and Science and leveraging our collections, going to different institutions, but also trying to collect fresh material. Fresh material is great for producing genetic data, particularly CO1 and these UCE sequences. So this work has taken me across many beautiful deserts, which I have absolutely enjoyed, from the Chihuahuan desert habitats in Mexico to Texas to the Sonoran deserts of Arizona. We mix techniques of using traps, but also trying to hand catch solifuges at night so that we can test them for their DNA sequences. The way that we wanted to go about testing these materials is first to produce a UCE whole family tree, and that is particularly in Ryan Jones' in preparation work. The goal of this UCE tree for me is to see if I can identify monophyletic clades that we could propose into being new genera. To do that, for the past couple of years, Ryan and I have worked together to sample either museum house specimens or a fresh collected material to generate genetic data. And so we use different kits depending on whether it's fresh or older material. Library prep was performed in-house using an Arachnida UCE probe kit so that we could target UCEs. Unfortunately, there's no genome published for Solifuges, so this approach helps work best for us to gather that information. We then sent out those prepared and amplified libraries to CU Anschutz so they can be sequenced, where we got those sequences back, cleaned them up, found those UCE loci, and produced a maximum likelihood phylogeny. That phylogeny then is the basis for us to produce generic hypotheses. Next, once those UCE libraries have been formulated, that's where my bioinformatics work came in. I wanted to use those CO1 sequences that we could generate from that data set to test species. As CO1 is known to be relatively well-performing as a species delimitation tool, that is the approach I took and wanted to use for these taxa. I used the MitoFinder program to assess the UCE libraries and pull out any CO1 sequences that it could find. From there, I took two different approaches. First, I formed a phylogeny, maximum likelihood tree, using those sequences in IQ-tree. I would align them, inspect, and clean them up using MAFT, ClustalX, and TrimAll to try to clean up those sequences and ensure that our branches would have good support. After that process, any sequences that had over 50% gaps were removed, as those were found to be poor performing in previous iterations. Once that phylogeny was set out, we could devise new species hypotheses. Next, we wanted to look at what the distance between those genetic samples were for each of those species. So we used a Chimera 80 distance matrix. Now, generally with this approach, there's no set guideline for a value on what creates a species, but lower variation values in a species is typically better support for saying that you should synonymize this or you should call this a species. Higher values tends to counter that and could potentially say that either there's a species complex here or you need to do additional approaches to delimit them. Once we have gone and tested the species within those genera, we wanted to figure out, well, what traits can we use to differentiate those genera? So we focused our morphometric approaches on a set of ratios to try to scale things. 267 individuals were measured, both from the Denver Museum catalog, but as well as many collections on loans. And what we first wanted to capture was a ratio we call ACP. This basically looks at how long the legs are compared to the body. Unfortunately, with the solifuges, when they're preserved, their bodies can shrink and crumple, so you can't necessarily use a total body length. So what we do is we measure the length of the legs and compare that against the head and propeltidium parts shown over here. And so those values tend to give us a sense of just how long the legs are compared to the body. Next, we also wanted to look at the chelicera because that was the basis of exploration that Muma started at, and we find a lot of characters there that we could explore. So for male and female chelicera, we generated a bunch of measurements, particularly focused on the total length and size, these two finger-like projections, looking at their size and how they scale, the position of dentition on those fingers, such as this medial tooth and this proximal tooth, as well as the relative depth and length of this depression called the fondal notch. For males in this group, they're all characterized by the retrodorsal process, so we also generated a length measure to see how it scales to the whole finger up top. For females, since there's less characters to rely on with the chelicera, they're more conserved in literature, and we know that. We also tried to explore the genital opercular plates. these ventral abdominal plates, which are diagnostic to females. So we generated 3 measurements following protocols from Erica Garcia's work in 2023. And we couldn't find appropriate ratios to scale this for, as the abdomen can shrink when the animal is preserved or for different reasons. So these are mostly raw. But for all these traits, we wanted to evaluate if they could differ the genera. So we used an independent samples t-test with the explanatory variable of genus. From there, once we could establish traits or ratios that differed these groups, we then looked at the species within the proposed genera. Could these same trait ratios that we're putting out then tell us a bit about the species there? Could we find things that distinguish them that are scaled? The goal of this was to use ANOVAs on these same list of traits using the explanatory variable of a species to try to provide diagnostic tools for them. If any difference was found, we then followed that up with a Games Howl test to identify exactly where the differences were between what species. So with all that hard work, I first want to present our UCE tree. And so this comes from Jones et al. In PrEP. And what we're finding, amazingly, is 2 monophyletic clades for the palpisetulosus species group that we sampled, a bit different from the 2015 findings. The first one I want to go through is the clade for nodularis, highlighted here in green. And so these are these Chihuahuan individuals, and they're quite morphologically distinct. Besides the fact that they're separate from other palpisetulosus, what we notice with the UCE tree is there's a nice split between these southern Chihuahuan lineages and these northern Chihuahuan lineages. Among the work and process with this too, we've also identified potentially new species, which we highlight here with the black arrow. And so they are morphologically distinctive, which we'll get into with the morphological results. The next clade I do want to highlight is what I call palpisetulosus, but this includes quite a few things. What we're finding with the expanded UCE tree is Ahuanis, those taxa from the 2015, are recovered now together with palpisetulosus, but also a few individual surprises as well. There are some California palpisetulosus taxa that are recovered in this group, and morphologically, at a quick glance, they seem to resemble and fit best with these species. But as they've not been reviewed, that is potentially for future work to explore. As for the species that we did review, what we find is a lot of mixture for the 1988 taxonomy that Muma and Brookhart put out. So a lot of this will need to be synonymized. In addition to that, we also have identified a new candidate species that we want to put out from the southern regions of the Chihuahuan Desert. What this tree has allowed us to do is target 2 new clades that we could potentially elevate into new genera, and that's exactly what we want to do to resolve this taxonomy. The first clade we want to name Eremobates, trying to keep that Eremo pattern in the names, but also reflect their blunt calicera morphology. 2, Eremodromus, again sticking with that Eremo naming scheme, but also reflecting the fact that these are lone desert wanderers, some of their biology in there. Next, with our Koan tree, what we found is we had good branch support after our bioinformatics pipeline and sequence, and that with the first genus, Eremobates, this helps us to identify potentially 4 species. It does contrast slightly with the UCE tree, where the UCE tree gives us a partition of a north and south split. This has more of a geographic breakdown. So this is the northern Chihuahuan individuals, and these are the southern Chihuahuan individuals, but the placement of taxa do differ a bit. In the next clade, Eremodromus, we highlight another 3 species as well. The first, Palpisetulosus, Bahade, and Ahuanis. The taxa that fall within those we find to be highly morphologically similar, and so synonymizing them may be a better approach. We do want to note that again within this clade, we see some discordance from the Co1 compared to the UCE tree, where the position of the California taxa and Ahuanis differ slightly. And there could be some reasons for that. It could be based on our sampling schema, whether there's gaps there or sequence quality that could affect the tree and where branches are falling. But there could also be biological reasons why UCE and Co1 differ. Co1 can be impacted by independent lineage sorting, the population structure of these animals, especially if there's a sex-biased dispersal. There could also be ancestral introgression altering things. So there are different reasons why the Co1 and UCE can contrast. At least for the species that we put out, we wanted to see, based on the genetic distance, does it seem stable to put these species hypotheses forward? So with the K80 distances, we looked at their values intraspecific and interspecific for the two proposed genera. What we find is generally the intraspecific comparing one species against another is comparatively low, which is good. We want that. That helps to give us evidence that likely what we put forward could be a novel evolutionary lineage. The exception is one species under Eremobatusis, Coelanus. We notice that with the sequences we produce, we get pretty high intraspecific values. So that could tell us two things. One, there could be an issue with the taxa that we're calling or naming, but it could also be any number of biological issues. Again, whether there's ILS there, independent lineage sorting, some population structuring. For us, we can use morphology to try to resolve these taxonomic issues, whether at the generic level or at the species level. So where we're at with the genetics is that from the UCE tree, we recover two separate monophyletic clades. One that we want to name Eremobatusis, and the other that we want to name Eremodromus. Co1 suggests synonymizing several of the taxa together, but there is some discordance with UCE, and that could be due to sampling, poor performing samples, or for biological reasons. Where we're at is we want to now determine what traits allow us to differentiate these two genera. So first, we looked at the ACP values, and what we did determine is that morphometric ratio helps us to determine that these two things are quite different at a glance. Eremobatusis males are reasonably smaller in their legs compared to their body than the palpisetulosus Eremodromus males. And within that, we also find that females too, that pattern still holds. The females tend to be smaller than males, but there is also that difference between the two genus. So we see a significant difference in genus, sex, and that effect between them. Next, the chelicera. Can we use that to differentiate between these genera? And the answer is, for males, yes. There are quite a few notable differences in the patterns of the chelicera, and I'll highlight some of them. One, for the most part, reflected in a lot of these ratios is that the Eremobatusis males tend to be a lot smaller and blunter, reflected by these two fingers. And so that pulls them apart away from the palpisetulosus Eremodromus males, which tend to have those much longer fingers. And a lot of the ratios there reflect it, such as the position of dentition on those fingers, where they are on there, and how the fingers scale to the entire chelicera. The exception where Eremodromus has larger values is in the fondal notch, that depression. Typically, they tend to have a much longer, narrower, but thinner fondal notch. The other one is the retrodorsal process. So typically, that retrodorsal process spans much more of the chelicera for Eremobatusis than it does for Eremodromus. With the females, though, at the generic level, we see a different story. The chelicera are more conserved, and so there are less traits that we can rely upon to differentiate these two genera. The exception tends to be that length-height ratio, which we can somewhat see within the picture here. So for the female Eremobatusis, they also, again, typically have much shorter, blunter chelicera than those with the Eremobatusis genre. While we did also explore the genital traits, we couldn't scale them at the time to any other component of the body, and so they might be skewed, even if the data says that there is a difference between the two genera. Particularly, that is visible in the position of the plates. However, these plates are hard to capture and accurately measure as they wrap around the abdomen. Next, we wanted to see, well, we know that the genera are different, but are the species within each genus different? And what we generally find is that for the first one, Eremobatusis, there's a problem, and it's that chelicera can't reasonably be told apart, at least from the perspective of these ratios. They're nearly all identical. Whether there's variance there, species doesn't best explain it. And so while the UCE tree calls for 3 species and the Co1 tree can call for 4 species, we think it's best to go with the more conservative approach and reduce down the taxonomy, as we just do not have the traits to currently revise the species. It could be a complex, but as it stands right now, we suspect that this Eremobatusis is best represented by 3 species. Eremobatusis guenini, the southern lineage's individual, Eremobatusis nodularis, which is distributed in the northern habitats of the Chihuahuan, and Eremobatusis guanajuatoanus, a distinct new species we sampled from the southern regions of the Chihuahuan, but actually possesses some additional accessory dentition on that lower part of their chelicera, and that is unique to the samples that we put into the phylogeny. For the females, there aren't traits that we could use, at least with the chelicera, to delimit species. However, with their genital plates, even though we find that the distance between them can be skewed based on not having ratios or the position and wrapping around of those plates, the shape itself does convey some information for us. Eremobatids guenini, depicted over here, tends to typically be placed together closer, and those anterior lobes are a bit longer. Whereas with Eremobatids nodularis, they're positioned typically further apart, and there's a bit more curvature and bluntness to these anterior lobes. Even though Eremobatids is hard to delimit with those ratios itself, but shape provides some information, Eremodromus is a different story. The males there are more easily discerned from their chelicera. There are a lot of ratios that we can use to delimit species, which we do so in a Games Howell post hoc tests. And as we go through the taxonomy, we can see some stark, unique characters, but also biogeographic signature. The first, Eremodromus palpisetulosus, is a Great Plains and North Chihuahuan taxa, and typically it tends to have a longer frontal notch compared to others, but also presents this weird feature, like a small narrowing ventral notch. The next is Eremodromus affinus or ahuanus. We are combining a species that we were not able to genetically review based on morphological likeness to ahuanus. And we find that typically the Sonoran lineage is among the blunter, more rounded, but also lacks that weird narrowing we see in palpisetulosus. It also tends to have the shortest RDP. Next is Eremodromus bahadae, which is the plateau lineage. And so this expands from the Colorado Plateau, Arizona Plateau, into the northern parts of the Chihuahuan Desert. And this typically tends to have the most sinuate, narrow, movable finger. And it has a frontal notch almost identical to palpisetulosus, but there are additional characters to the body that we can use to delimit it. Next is Eremodromus norrisii, which we could not generate Co1 samples for, but the UCE tree says it could be a stable species. And this typically has the smallest frontal notch in the group, as well as the most prominent ventral notch. As for other characters, it tends to fall in line or be identical to other palpisetulosus species. This one tends to be distributed across the Chihuahuan Desert, but does make its way into the Great Plains. The last one is a new species that we recovered with our UCE data, peludo, and it has some of the most distinctive morphology with the chelicera, and this is also from the southern regions of the Chihuahuan Desert. We suspect that there could be more new species described to this genus, but we're still waiting on some more genetic data. With females, again, similar to Eremobatids, it's hard to differentiate the females, at least by the chelicera. For the most part, they are nearly identical to each other, with the exception of that affinus ahuanus species that we're lumping together. It tends to be the smallest female, reflected in cheliceral ratios. The plates, though, are a great resource for delimiting the species. Though we can't capture measurements at this current time to delimit things, the shapes tend to be quite unique. While they're all broadly triangulate and ending with anterior lobes, the shapes of the lobes, the shapes of the plates, how they curve, tend to differentiate between these lineages, and so that helps provide new information that we can revise these species with. So overall, our phylogenetic and morphometric analysis hopefully have paved the way for a taxonomic revision of the palpisetulosus species groups. This work has highlighted the fundamental use of combining genetic data with morphological data. Co1 data in itself is a great resource for species delimitation, but it can be subject to many different pitfalls. And so it's best paired with either additional genetic data, like having that UCE backbone, or comparing and contrasting it to the morphology of the animals that you work with and review. In this case, the Co1 contradicts the UCE tree, and that could be due to reasons of limited samples, gaps in our phylogeny where we have still biogeographic gaps, but it can also be due to many biological reasons seen in different publications, where it's either ILS, ancestral integration, and so ancient Wolbachia infections, many different things. We hope that at least combining this genetic work with morphological work has provided a new and robust taxonomy. And at least we've highlighted the power of morphometrics to differentiate the proposed genera that we are reviewing. And along with that, provided new diagnostic traits for species that we think can last much longer and be well more supported. But there is going to need to be additional integrative, morphometric, and genetic analyses in the future. There are still gaps, there are still trait limitations, and we hope that this foundation, this work, sets the way for someone in the future to follow up and revise the taxonomy again. Thank you.
Time for questions, and you can ask anything about Solifuges.
First, I want to say that was a really great presentation, and you obviously seem very, very prepared. And so I think you're going to do great on Friday. I have two questions. First, and I wrote them on my phone, so I'm reading them off. Given that you focused on the CO1 gene differences for the species delimitation, and that's mitochondrial DNA, would you consider incorporating ribosomal DNA to complement your findings and potentially offer a broader perspective on the differentiation of the species?
So that is a great question. And just to reiterate, Bridget was asking, besides using CO1 DNA, which is mitochondrial, can it be complemented with ribosomal DNA? Currently, I don't know how that would look for solifuges, but I've seen it applied in papers. I don't know off the top of my head what answers I could get or what that looks like, but I think it's potentially in the future something that can be explored, especially as now there is genetic work occurring with solifuges. A decade prior, the work I do would not have happened. Paula and her team set the foundation for that.
Okay, thank you. My other question is, what are the geographic factors that might be contributing to the differentiation of the species, given that they're in the different parts of the area? Like, is it primarily the physical distance between the populations that you think might be driving changes, or are there other environmental factors that might play a role in the divergence?
Okay, awesome question again. So Bridget was asking that for these species that we're calling, what could be contributing to the speciation? So is it something geographic? Is that the pattern that we're seeing? Or could it be another climatic variable potentially? My answer is it could possibly be both. So one thing to note about, which I don't get into the details of my research, is that with both the 2015 and also some ongoing analyses, this is one of the latest diverging groups of the Eremobatids, so only in the past million years. They're likely still radiating and expanding, but there's potentially, I think, some barriers that we can identify geographic, which are keeping things split. So, for example, the species that distribute in the Colorado Plateau, we typically only see one individual there, and others aren't making their way into that habitat. They're not getting past those mountainous zones, but it's interesting to see one lineage, for example, do that. Am I confident that that could be the barrier? No, I think there'd need to be additional work to see what exactly is keeping these taxa online. I think climatic variables can also explain some of the distribution patterns. So some ideas I've thought of is whether it's the temperature of the soils, since that's where they spend most of their life, humidity, average temperature in a year, the rainfall, so different things.
Okay, thank you.
Other questions? And it's fair to ask just sort of general biology questions of Solifuges.
I have far easier questions than Bridget did. Wonderful job. And I know for certain, like mammals and birds, there's ways of kind of live trapping and keeping an animal alive, wing clips, things like that. Are there ways to do that with Solifuges, or does it have to be like trap and kill?
Also a great question from Tiffany. So Tiffany asked, with other animal taxa, typically there's situations where they can capture, keep things live in the lab. And is there ways that we can do that, or does it have to be trap and kill? And for the most part, trap and kill to guarantee you have the animal. I have tried in 2 situations independently to raise juveniles into adults, and about 1 week later, they disappeared into the dirt and never existed again. We have not found a way to raise them, but I've heard from hobbyists, I've heard from undergrads who've been in our lab that they've gotten things to live for a few months, and in some cases a year. And if anyone has answers, I always welcome them to tell me, because I would love to raise them.
Okay, and second is sort of a more general question. You are proposing, some new taxonomical changes. How do you actually go about, from your proposal and publishing a paper to creating that change or getting that widely accepted?
That is another good question. Tiffany asked, from the paper that I'm putting out, this proposal, how do I go about publishing that and then putting out this new taxonomic change? And to be honest with you, where I'm at right now, I'm still learning the process, so I do not know all the steps. But I know that the first step I want to accomplish to do that is get this into a publication where it can be reviewed and formalized. I know this has to be submitted past that to accept that these are new species. I know I have to submit the sequences that I produced to also have those accepted, but parts of it are still dark for me.
Other questions? John, you got anything that this committee might ask? Yeah. No. Other questions? Sure.
Oh God, no, I'm kidding.
No, this one's going to be easy. I just want to know if there's anything that you are prepared to talk about that wasn't in your presentation, that's just a cool factor of your research that you want to talk about and didn't get to.
Quite a few things, and that was the problem with exploring the taxonomy. So just a step back, Bridget asked if there was anything that I didn't present in my talk today that it was work I'd done that I really wish I could talk about. And my answer is there's quite a few things. There's so many traits I've tried to explore to stick to the wall, and a lot of things that I thought would work just fell through. Like the pedipalps, those front appendages they use, they seem to be sensory systems for them, and there's characters that pop up there. Could those consistently work? And they fell through for me. I couldn't find patterns there, but in other clades it seems to work. This palpisetulosus group, I now understand why Muma and Brookhart threw things to the wall, because there seems to be consistent patterns and then you find exceptions. Another system I looked at was called Ctenidia. It's like these combs of hair on their abdomen, and there seems to be patterns there which can help and delimit some species, but then in other situations you see so much variation, it's kind of just, you gawk at it and go, what do I do with this? The other thing is I also looked at phenology, like what are the collection times that we have for them, and can that inform us of something? And almost consistently, my taxa are just summer species. So I think that doesn't really help to separate them, but that's also a good argument for synonymizing things. If they're in the same habitat and they're out at the same time, there's probably a good chance that there's some gene flow.
Okay, thank you.
So sorry. Any other questions? Oh.
So with these guys, I've known this for a long time. You look at characters on the chelicerae. I know there's not a whole lot known about the biology of many of these species, but is there any hypotheses as to why they have the variation in the chelicerae? Does it have to do with prey, different types of prey, or is it just variation, maybe non-selective variation that just accumulates? In other words, can you look at this and go, this one specializes on these kind of insects or other invertebrates, this one is more this kind?
That is an excellent layered question, and I'll try to tackle this as best as I can. So John was asking, looking at the chelicera, what the variation do we see? How do we necessarily know that some of these traits are being selected for, that they're special in that sense? So first, because the Solifuges are using these chelicera for mating, and we see such variation there, that's a good insight that there is potentially selection for some of these different characters, and we see consistent patterns in different clades. Unfortunately, there is also a lot of convergence for these traits in the whole family phylogeny, but as for what's being selected for in certain groups, that is difficult to say, especially without knowing their function. A lot of these traits, we still don't know exactly what they're doing, despite the fact that they mate with the chelicera, like the retrodorsal process. I could only presume it may have something to do with an interaction of the chelicera and the genital plates, but there's no way to know without visualizing that or further testing. And the other thing is, I can't positively say what's being selected for or not, without much more genetic detailed work, like looking at raising generations, and that's not somewhere we can be at this current time. So thank you.
Are there questions? All right, let's thank Goran.